Okay, so this is the first video looking at proteins, and what this video will look at is just the general structure and probably just the first half looking at the basic building blocks of proteins before the next video builds that up further into the more complex structure. You can see just even from this intro slide, so these are all pictures of different types of proteins, and even though they're fairly complex, I suppose that's the main thing I want you to understand from the get-go is overall protein structure is really complex. They're very specific, and we're not talking about simplified bonding and so on. So, but what we will look at is, I suppose, how we can simplify the type of bonding that does occur, even though it is fairly complex. Okay. So we're going to start. I will just take you through the study design. In terms of the amended study design, there's nothing in terms of protein structure that has been amended. Okay, so in terms of your questions in your book and so on, everything will be applicable for your SAC and for the exam. Before I actually look at molecule structure and so on, what I want to just go through is what proteins actually do, because it's important that you understand just a little bit around protein function. Most people generally tend to go along the lines of proteins build muscle, and you eat more protein makes you stronger, that kind of thing. And in a sense, that's one function of protein, but it's certainly not the most important function. Generally, you can categorize proteins like the different types here, but just be aware that we are looking at a huge, broad function for the human body. And if you think about the function of what different parts of your body do in terms of bone and muscle, cartilage, enzymes, hormones, well, they're all structurally different. And so that's what we'll talk a lot about over the next week, is how our different proteins are structured differently to enable them to perform their function as well. For those of you that do Year 12 biology, this will sort of enable, help even some of your understanding of enzymes and so on that you would have already done. In terms of the type of protein, the only type that we will look at in great detail is an enzyme, and that will be predominantly what we'll be working on next week. Over the coming lessons, okay? So we'll talk a lot about enzymes and their function and so on. Okay, so this video particularly, what we're going to look at is the general structure and the basic structure of what makes a protein. And so I like to think about proteins as kind of like, if I was to make them really simply put, I was going to think about a necklace with lots of beads all joined along, okay? So the reason for that is that the molecular structure of a protein is kind of a little bit like that. Okay, so if you were to look at the molecular level of a protein, what we will see is these small molecules called amino acids, and it's lots of these amino acids joined together that make up a protein. That's kind of what you need to visualize at the moment. Obviously, they're not circular shaped; they're much more complex in shape. But just in terms of a basic understanding, just think about that for the moment. So to start with, we're going to look at an amino acid, what it is, what the general structure is, and so on. The picture here gives you the general structure of an amino acid. So there are actually 20 amino acids that make up all of the proteins in the human body, and they all have a very, very similar structure, which is really helpful and good. So you'll notice that they have this central carbon, and it's called an alpha carbon. On one side of the central carbon, there will be an amino group, which we've looked at an amino group before. It's a base group. It acts like a base. It can accept a hydrogen ion, and it will have a carboxylic acid group on the other side of that carbon. Carboxylic acid group or carboxyl group behaves like an acid, can donate its hydrogen ion very willingly. It will have a lone hydrogen on the other side, and in our fourth covalent bond, there will be another branch or another side group or another cluster of atoms that will be varied. Okay, so it's this side chain, this R group, or sometimes it's seen as a Z group, is what is different or what is varied for each amino acid. And it's what will determine the structure of the overall protein and absolutely a function of the overall protein. Okay, so you will hear them referred a lot as alpha amino acids, and as I said, that's because we have an alpha carbon here, and it's because our amino and our carboxylic group are attached directly to that alpha carbon. There are slight differences in other types of molecules, but we're not getting into that. We're just looking at alpha amino acids. As I said before, there are 20 of those, and so it's the combination of these as they bond together and join together that create all of our different types of proteins. Out of 20 of those, 9 are what we call essential and are required in our diet. And as you can probably see from the overall structure, we've got an amino group, which is polar, and we've got a carboxylic group, which is also polar. So no matter what's on the side chain here, all of our amino acids are soluble in water. Okay, some will be more soluble depending on the side chain present, but they all are soluble. And because your amino group here and your carboxylic acid can form hydrogen bonds with water. They have intermolecular forces required to dissolve within water. Some other examples. So a really good thing to do right now is to download the Databook from either VCAR's website or from OneNote and have a look on, they start on page 14. So all 20 of the amino acids are actually listed there. You'll see the name. Oops, I'm going to get back onto my pens. You'll see the name and you'll see the symbol. Okay, and we use the symbol, obviously it's just a quick way of referring to the protein, and we can use that as well to show a protein sequence as well. You will see the structures there. You won't see this coloured highlighted bit. This is highlighted bit is clearly just marking those different R groups. And you'll see them generally shown exactly like this. It's a semi sort of condensed structure. The good thing when we're drawing macromolecules from this point onwards, we don't have to worry about drawing every bond or anything like that. You can kind of move between full structure and condensed structure. You won't really get marked down at all in the exam. I generally will instruct my students to copy the structures very basically as they are shown in the Databook, which is effectively a condensed structure that just generally will show this bond, and this bond. But of course, you could show the hydrogen bond, that bond there between the carbon and hydrogen as well. There'll be no, you won't, I think you will get marked down for anything like that in the exam or in your SAC. So these three just examples of some, alanine having a very, very simple side chain of a CH3, and arginine much more longer. It's got a double bonded carbon to a, not really an amine, an NH group, another amine group there. And you've also got asparagine. Some of the names are interesting. Slightly different again, okay? And I will explain it probably in the next video why these side chains are so important. Okay, so we're just sticking with simple, just basic amino acids for the moment. Now, if you do have a single amino acid in solution, they won't actually appear as molecules. So what I mean by that is they're actually in a neutral solution or in any solution, they're not going to actually appear like this. This is how they do initially bond together in terms of overall covalent type bonding. But because of the presence of so much dipole within these molecules, because we've got this amine group and because we've got this carboxylic acid group, what happens is these amino groups and the carboxylic acid groups behave as they normally would. They behave as a base and they behave as an acid, because an amino group is a base and a carboxylic acid group is an acid. So when placed in solution, particularly within, obviously, we're thinking about a watered solution, a living organism, so think about the human body, a large amount of water present, well, they are going to behave just as they would if we were reacting them with water in the lab, okay? So if I might just write it on here, if you have an amino, think about amino reacting with H2O, think about this just chemically before we take it into the molecule, that this is the base, and then water would automatically behave as the acid. So the water donates a hydrogen ion, and the amino accepts, oops, I've done too many hydrogens there, NH2, NH3 positive, okay? So remember, water can behave as both an acid or a base depending what it is reacting with. And in this case, NH2 is a fairly strong base and will therefore mean that water inadvertently behaves as an acid, okay? Same thing if we take, I'll just write the functional group rather than the full acid, carboxylic acid and water. Obviously, we've got acid there, and water inadvertently acts as the base. The acid will behave and donate the hydrogen ion, and that will be accepted by the water. Okay, that's generally what we know in terms of overall acid, base, and water, and so on. Okay, but what happens in terms of an amino acid is that because now we have a really strong base present in solution and we have a really strong acid, the water won't act as an acid and the base, and the water won't act as a base because we've already got a strong acid and a strong base in solution. So they still act. I mean, the amino group will still act as a base. It will still accept the hydrogen ion, but it won't accept it from water. It accepts it from the carboxylic acid. Okay, so we move from this neutral molecule here, okay, this molecule here, and move it over to here, where our carboxylic acid functional group has lost a hydrogen ion, hence the negative charge there. But that hydrogen ion was accepted by the amino functional group on the zwitterion and has gained a hydrogen, so it forms an NH positive, NH3 positive. Doesn't matter where you put charges as long as they are associated around that functional group. That is the main thing. Okay, so in a solution, we see this behavior occur all the time. Now, this is an example of in a neutral solution, so like a pH of 7, basically. So as we start to change what the solution looks like, the behavior of the acid and base can also change, okay? Okay, so I'll use this example to explain that further. So we take this, oops, I've just moved that. That's not what I want to do. Here we go. This molecule. So this is the zwitterion. This is the molecule we just established before that is behaving as an acid base with the amino and carboxylic acid functional groups on that molecule. Now, if, for instance, we take this molecule and we place it, or we put it in a beaker of hydrochloric acid, okay? So it's present in a very low pH, all right? So think about it. There's lots of hydrogen ions present within that solution, okay? What that then means is because we're at a very low pH and in the presence of a strong acid, it's once again what I was saying before. What happens inadvertently is because we have an acid present, the acidic function of the amino acid won't take part because I suppose think about it, it's trumped in a sense by the acid in solution, and the amino acid will instead just behave as a base and accept the hydrogen ion. But it's from the solution and not from its corresponding acid on the molecule, not from the corresponding carboxylic group on the molecule. Okay? Now, I suppose there's argument to say, well, what if the pH is around 6 or 5? What would happen? And it would be fairly difficult to actually determine where's the hydrogen coming from. Is it coming from the acid in solution, or is it coming from this guy here? But you're not going to be tricked in that sort of sense. We are going to be looking fairly at the extreme. We'll look at the neutral pH of 7. We will then look at a fairly low pH. Okay, so when in a solution of acid, the amino acid then behaves inadvertently as a base, and we form the NH3 ion here. We take it to the other extreme, ion at high pH. Okay, so the amino acid is placed in a basic solution with lots of hydroxide ions present, very willing to accept hydrogen ions. What that means is it inadvertently acts as an acid. Okay, so acts as an acid. This amino group gets sort of trumped, doesn't actually end up involved in the chemical activity, but it's the acid component, and we would still have that negative charge there. Okay. So what happens there, therefore, is we don't have a zwitterion anymore. A zwitterion is represented by this middle molecule here. All right, technically is overall neutral charge, but has charges present. When we change the pH of solution, the behavior chemically can obviously change to either acting as a base or acting as an acid. Okay, the function of this in terms of the human body is actually quite vital as amino acids behave as what we call buffers. And so what that means really, really simply, because we're not getting into this too much, is that they have an ability to accept hydrogen ions or accept hydroxide ions and behave like an acid and base and minimize the effects of high concentrations of these, which if in the human body you alter the pH out of an optimal range, you can get very, very sick very, very quickly. Okay, so the human body has a very, very specific optimal range in terms of pH and temperature, and we can't go out of that range. So buffers within the body systems ensure that we keep our body within that range, particularly if you consume something or if you're exposed to something that shifts your pH out of optimal range. Okay, just a couple of practice questions just to finish off. So it's a nice, fairly complicated amino acid histidine. Definitely have a look at some of the amino acids in the data book. There's some interesting side chains there, so you just don't get thrown by them when you do see them. But in terms of structure, this is just a cyclic molecule. There's an amino group on the cyclic molecule. You can see there. I'll get my pen back, okay, or not an amino, an NH group, sorry. Not quite an amino. You've got a cyclic molecule. It's just a slightly different side chain. Question, draw the structure of histidine at pH of 2. So at a pH of 2, so therefore we know the solution is an acid, okay? So what will happen therefore? What will this behave as? It's going to behave as a base. And I want you to draw, I'll pause the video and I want you to draw what that would inadvertently look like. Okay. All right, so I'm going to draw the easiest sort of way to tackle these guys. I tried to draw the central carbon first. I'm going to probably put not all the bonds, but these main bonds in. So we're not going to touch our acidic functional group because it's acting as a base, so it will accept hydrogen ions. So we're going to look at the base functional groups, okay? So this guy here, so we've got an amino group. It will act as a base. It will accept a hydrogen ion, so NH3 positive. You can draw it out in full, that's fine. We're going to finish drawing our side chain. Not very good at drawing these on the computer, sorry. N. Now, what will happen, do you think, to this functional group? It's an NH. It's technically not an amino. I can't remember the name of it. But it will also behave as a base. It will accept a hydrogen ion, just like our one over here, this one over here, and behave as base, particularly if it's in a situation where it's at a really, really low pH. So we must have a fairly strong and or fairly concentrated. You definitely need a strong acid to get a pH of 2. If you're ever confused about that, generally, if you know that there's 2 base groups, they both will ionize and accept hydrogen ions. And in the same sense, if we're in a really, really high pH, if we have 2 acidic groups, same sort of thing occurring as well. So that will take me to, yes, our couple of other practice ones here. So really good, use the data book and just have a practice of drawing these and drawing the amino acids in the different pHs. All right, so have a pause of the video and I'll do the answers. So serine at a pH of 7. So this would be what we would call a zwitterion, okay? If it's in solution, you know it's at a pH of 7. So you have a look at the structure of serine. I might draw it up here. Oops, now I've just lost my pen again. There we go. Carbon, hydrogen, COO negative, NH3 positive because it's a zwitterion, CH2OH. Okay, so it's ionized both at the acid and the base. Okay, that's that one up there. Asparagine, pH of 1. If it's at a pH of 1, it will be acting as a base Okay, so it'll be accepting hydrogen ions. So carbon, hydrogen, leave the carboxylic acid functional group. We've got the NH. Now it's acting as a base. So this basic group will accept hydrogen ions, forms NH3. Side chain CH2. Oops, that was odd. C, double bonded oxygen, and we've got another NH2, which will also accept hydrogen ion and act as a base in a very low pH. Leucine, last one. We have central carbon modded to a hydrogen. It's at a pH of 10. So it's in a basic pH, which means it will act as an acid. Okay, acting as an acid. So we're going to leave the basic functional group alone. Its carboxylic acid functional group here will act as an acid. So it will donate the hydrogen ion and form a carbonyl COO negative. And then we'll have our side chain, which, oops, I've run out of room a little bit. Have only CH groups, so no acid or base properties there, and we can leave it just as it is like that. If you want to draw them out fully with showing all bonds, no problem with that. You don't have to draw them exactly like I have done. What I'll do there is just leave it there and finish off the next half for next lesson.